Now let's see again. For example, we have taken this a sensory neuron. This shows a sensory neuron from a giant squid. Why we have taken this from a giant squid? Because it has a wider diameter, and we can easily insert this electrode inside this sensory neuron. Okay, so we see this is sensory neuron, which has this myelin sheath present on outside. This is a myelin sheath present on outside at intervals. Okay, so this is myelin sheath. We have this myelin sheath as well. And to see the potential difference between inside and outside of a cell, we use this millivoltmeter. Okay, this millivoltmeter has two terminals. One terminal is placed on outside this myelin sheath here. Okay. Sorry, what did you say? Okay, so and the other terminal or electrode is inserted inside the nerve cell or into an exoplasm. The potential difference between inside and outside measured when this nerve cell is at a resting state is known as resting potential. Okay, so this is resting potential. We can define resting potential as a potential difference of minus 70 millivolts between inside and outside of a nerve cell when it is at a resting state. Okay, we can plot this on a graph showing, if this is, for example, 0 millivolts potential difference, so resting potential would be minus 70 millivolts. So this is minus 70 millivolts, and at a resting state, this would be potential difference between inside and outside of a nerve cell, and this is known as a resting potential. Now the question is how this resting potential, like this potential difference between inside and outside of a nerve cell, is maintained. The inside is negatively charged and outside is positively charged in this case. Okay, so it is positively charged on outside and it is negatively charged on inside. So the question is how it becomes negatively charged on inside and positively charged on outside, how this resting potential is maintained. So let's see again. How is this resting potential maintained? So to understand this, let's see I draw this an axon again. So you see this is an inside and this is an outside of an axon, and it has myelin sheath here. This is the myelin sheath present here. This is myelin sheath present here. This is myelin sheath present here, and we have myelin sheath present here. Okay, and there are nodes of Renvier. If this is myelin sheath, you see this is node of Renvier. Okay, so at these points, at these nodes of Renvier, there are sodium potassium pumps present. This is sodium potassium pump. Okay. These sodium potassium pumps are active at a resting state, and they remove 3 sodium ions out of a cell by inactive transport, which are replaced by 2 potassium ions into the cell. So if this continues for a while, the sodium ions are removed out of it and the potassium ions are taken into it. So what would happen? Outside it would have more positive and inside would have less positive, which is represented as negative on inside and positive on outside. Hope you have understood it. Okay. So this is how this resting potential is achieved. Now, the transmission of an action potential, or how an action potential is produced. Let's see. I'll draw that again. So let's see, this is an axon or a dendron from a sensory neuron taken again. This is the inside, and this is the outside of a nerve cell. Okay. So initially it has positive charge on outside, and it has negative charge on inside. Okay. And if we apply an electric current here, okay, so this is electric current applied here. So we do an experiment to see how the potential difference would change between inside and outside of this nerve cell if an electric current is applied here. So we apply an electric current and it opens channel proteins here known as sodium channels. Okay, so these are other transmembrane proteins other than the sodium potassium pumps. So sodium potassium pumps present here, they become inactive. These are sodium potassium pump. They become inactive and sodium channels open. Okay, so consequently, as you already know, during the resting potential, there were more sodium ions pumped out of the cells, and there was high concentration of sodium ions outside, and there was low concentration of sodium ions on inside. Okay, so consequently what happens when these sodium channels open, sodium diffuses into the cells. Okay, so if more and more sodium diffuses into the cell, inside of the cells start becoming positive. As more and more sodium ions enters in, inside becomes positively charged, and outside becomes negatively charged. Okay, why does it happen? Due to influx of the sodium ions, and this is known as depolarization. This is known as depolarization. Okay, if we record this change in the potential difference between inside and outside of a cell with the help of an oscilloscope, and we record it on a graph paper. So as I said earlier, if this is the graph showing 0 millivolts potential difference here, and this is initially minus 70 millivolts here, which is resting potential. And we opened, this is where, this is time on x-axis, and this is the stimulus provided here. Or you can say electric current applied here. Which opens sodium channels. Okay. So if sodium channels open, sodium would diffuse in. And if more and more sodium diffuses in, this potential difference becomes, less positive on outside and more positive on inside. And this would be drawn as, graph rising up to the, so it would first turn 0 millivolts, and then it keeps on moving ahead up to plus 40 millivolts. Okay. So sodium channels are open here. Sodium channels open. So sodium keeps on moving in, and this potential difference that reaches to plus 40 millivolts. Until it reaches plus 40 millivolts, the sodium channels don't close. So this change in the potential difference is known as depolarization. This is depolarization. Okay. When this reaches at plus 40, sodium channels close and potassium channels open. At this point, sodium channels close and potassium channels open. So when the potassium channels open, potassium goes out of the cells. As you already know, here during the resting potential, there were sodium ions being pumped out of the cell, and potassium ions were being moved into the cell. So as a consequence, there was high concentration of potassium ions on inside, and there was low concentration of potassium ions outside. So consequently, when the potassium channels here open, the potassium goes out at this point. Okay. So as a consequence, this potential difference that again returns to minus 70. Okay. So as potassium channels are open, so potassium is moving out. So this potential difference that returns to minus 70. This is now known as repolarization. Okay. This repolarization doesn't stop at minus 70. Instead, there is a little overshoot. No, it goes little below this minus 70. And then only the potassium channels close. At this point, potassium channels close. Okay. And then this returns back to, minus 70, which is again maintained at a resting potential by the sodium potassium pumps becoming active. So this overshoot little below this minus 70 is known as hyperpolarization. Okay. And this is resting potential again. So this wave of depolarization, repolarization is known as an action potential. This is an action potential. So we can define this action potential as action potential. A wave of depolarization and repolarization produced along a nerve cell is known as an action potential. Okay, and this is divided into depolarization, two parts: depolarization and repolarization. Depolarization is when inside becomes positively charged. Inside is positively charged. And repolarization is when inside is negatively charged again. Okay, so inside is positively charged. Why this is positively charged? As sodium channels are open and sodium diffuses in, so this brings membrane to become depolarized, and potassium diffuses out. Diffusion of influx of sodium ions that makes the membrane depolarized, and the outflux of potassium ions that makes it repolarized. So let's see, we have some questions here. This is, for example, figure 2.1 shows the changes in membrane potential in an axon during passage of a single impulse. Okay, so this is time on x-axis, and we have the potential difference on y-axis in millivolts. Outline how the resting potential from A to B is maintained. So you see, this is here the resting potential from this point to this point. So this is resting potential. Okay, we discussed that sodium potassium pumps become active. Okay, sodium potassium pumps are active, and by active transport, which is use of ATP, and against the concentration gradient, 3 sodium ions moved out and 2 potassium ions moved into the cell. Okay, so this is how this resting potential is achieved. Now we have this from B to C. This is depolarization. Okay, and why this depolarization occurs? If at this point, sodium channels open, sodium will influx, and inside would become less negative or more positive, and outside would become less positive, and this is how it becomes depolarized. So from this point to this point, sodium channels are open and sodium moves in. At this point C, sodium channels close and potassium channels open. So potassium from this point to this point goes out. Okay, potassium moves out, and in this case sodium moves in.
Can you explain it again?
Okay,
Yes, sir.
from this point B to C, potential difference, you see, is becoming positive on inside and negative on outside. During this stage, during the resting stage here, from this point to from A to B, potential difference inside, this nerve cell was negatively charged on inside, and it was positively charged on outside. And this potential difference is maintained due to insulation which is provided by the myelin sheath here. Okay. And I explained it. How does it happen? As 3 sodium ions are moved out and 2 potassium ions are moved in. So you see there are more positively charged ions being moved out and less number of positively charged ions are being moved in. So consequently, outside become positively charged and inside become negatively charged. So this is how resting potential is achieved. Okay, at this point. Now, if the sodium channels open and sodium potassium pumps close at this point, okay, this is myelin sheath here. And if sodium channels open here and these sodium potassium pumps that were active at resting stage are closed now, or they are inactive now, so what would happen? These are sodium channels, they open. The sodium would diffuse in. Okay, so if more and more sodium diffuse in, inside would become positively charged and outside becomes negatively charged. So this becomes more and more positive. So this graph is, you see, this is going up. Okay, this is going up to plus 40. So inside becomes positively charged and it reaches to plus 40 millivolts. And this is known as depolarization. When this reaches at plus 40, sodium channels close and potassium open. So if potassium opens, potassium channels open, already, you know, at this stage, potassium was high concentration on inside and potassium was low concentration on outside. Okay, this was low concentration on outside and high concentration on inside. So consequently, what happened? Potassium that diffuses out. So if potassium diffuses out, positive charge or positive ions go out of the cell. So inside becomes, you know, inside becomes this negatively charged again. And this returns back to, you know, the minus 70. So this is known as repolarization. So this is repolarization. Okay, and this doesn't stop here at this point. Instead, it goes little below this minus 70 up to this point, and this is this overshoot or this little lower potential difference below minus 70 is known as hyperpolarization. And when this potential difference reaches below minus 70, potassium channels close here. And sodium leaks into, and that returns back into up. And then this is maintained, you know, sodium potassium pumps become active again. And this potential difference is maintained at minus 70. So this is the resting potential achieved again. Hope you have understood it. Okay. So let's see. This is an AS question actually. Figure 1.1 is a diagram of a part of a neuron membrane at resting potential. Can you identify with reference to figure 1.1 name A, B, and D? What is an A? What should be this B? What you think should be this D? And what should be this C? Okay, as I said, this is sodium potassium pumps. You already know these are integral proteins embedded in the phospholipid bilayer. So A should be sodium potassium pump, or maybe you can write carrier protein. Okay, And here you see there are 3 molecules represented here which bind at this site, and they are moved out of the cell. So this should be the sodium ions. And then here you see this is 2 molecules shown here. So this should be the 2 potassium, and this is 3 sodium. So sodium moves out and potassium moves in, and this is an this is by an active transport where ATP is used. So this must be this C which binds here and brings about a change in the conformation or the shape of this protein. This must be an ATP molecule which is used to bring about an active transport. Okay, so this is how we can identify these different components. Explain why maintaining resting potential that requires energy. Obviously, we discussed maintaining resting potential moves the ions against the concentration gradient, which is an active transport. So for moving these ions, sodium ions, out of the cell against the concentration gradient and potassium ions into the cell against the concentration gradient, energy is needed, okay, as it is an active transport. So ATP is used in this process. Describe how the changes in membrane bring about depolarization from B to C. We have discussed reference to the electric current that we have applied. Sodium channels open, sodium diffuses into the exon, and this makes an inside positively charged and an outside, and the potential difference across or potential across the membrane changes. So that is what is known as depolarization. And similarly, repolarization explain how membrane is repolarized from C to D. Reference to sodium channels close, potassium channels open, so potassium diffuses out. And if more and more positive charge on the potassium that goes out of the cell, it restores the membrane or the resting potential. It returns back to -70. But there is a little overshoot or hyperpolarization, or when this hyperpolarization takes place, only then these potassium channels close, and then the resting potential is achieved again. The sodium potassium pumps become active and resting potential is achieved again. So we can draw this in action potential like, this is 0 millivolts on a graph, and this should be the resting potential here. This goes up to +40 here and returns back to this -70. So this is -70, and this goes little below and then returns back. So this is resting potential achieved again. So this wave of repolarization is known as an action potential. Okay, and a period from this resting potential to restoring the other resting potential is known as a refractory period. Okay, so we'll discuss how we can find a refractory period from a graph and what is the importance of this refractory period in the next lesson. G, anything else? Is it okay? So let me mark your attendance.
सर
जी
सर मेरे फोन से डिस्कनेक्ट हो गया था। आमना मैं वीरा के साथ ही रह रही हूँ।
Okay. So that's it for now. आपका टेस्ट जो है वो होगा